All right. Good afternoon, everybody. Welcome back. Hope everybody had a fantastic, good morning, good afternoon. Hope everybody had a fantastic weekend. Hope everybody has a fantastic class today. Does anybody have any questions before we get going? Any concerns, clarifications needed? I'm going to give you think about that for a minute if you've got a question. I got a couple of questions about the Google sheet we worked on last class, the Chargaff's rule one with the HTLV sequence, %A, %T, %G, doing some calculations on Google Sheets. Didn't need to be turned in. A couple people were wondering if that was an assignment. Most of the things that are in the course manual as exercises, most of the things I will not ask you to turn in for points. They're practice. They're like homework assignments with no points attached. If you want to practice them on your own, do so before class. Come to class, we'll talk about the answers if you're interested. If you ran into a problem, we'll talk about it, but they usually won't be due. So if you don't want to do them, you don't have to do them. Your grade might suffer if you're not practicing. I just don't put points usually behind that to motivate you. I haven't graded all of the surveys that I asked you to take yet, so if you haven't seen those grades pop up on Canvas's grade book yet, that's why. Sometime this week I will go through and score all of those. Those are just for completion, not for the actual answers. And for the few of you that have asked about Socrative maybe not recording your responses when we've done these little start of class, end of class activities, we're going to do one at the end of class today. And if you want to come up as soon as you're done with that and check with me to make sure that your name and your answers got recorded today, let's do that so I can assuage any fears. Final thing before we get going is that, has anybody, let's see, can we do a show of hands, please? Who has downloaded the slides for today and is looking at them now or has at least looked over them? All right, excellent, most of you. Have you seen how long they are? I do not intend to get through all of this today. So you should never come to class, please, don't come to class assuming that I'm trying to power through everything that I have prepared. I've prepared extra. So I'm happy to go at whatever pace the class wants to go at. The schedule of the class and the syllabus has got plenty of room padded to make sure that we can go and talk about things that you want to talk about if there are confusion or if there are other things you want to know about. I'd rather be over-prepared than under-prepared, in other words. So any questions at this point? All right. So this is a little bit of stuff that I wanted to talk about at the end of last class, but I didn't get time. But again, the point of these is to give you a little bit of sense of how everybody in the room feels about their abilities in genetics. This was from what's called a knowledge survey, and this is what last class I told you, these sorts of questions that I was asking you, the specifics of the questions were basically what I'm going to ask you to do on exams. Can you label nucleotide polarity on DNA, those sorts of things. And I was just asking people anonymously to rate their confidence and their understanding of that, whatever the topic was. So there were 58 of those questions. Thanks for those of you that waded through all 58 questions. And I just wanted you to know that on a scale of 1 to 3, the maximum that anybody in this room rated themselves at on any question of all of those 58 questions, the top score on average for each of those 58 questions was 1.4, meaning about half of the class said, yeah, I can do it, and about half the class said, yeah, I'm pretty confident I can do that. So there are some topics in class, in other words, that were on that survey that everybody thinks they're pretty good at already. And this is helpful for me because then I know how much time to spend in class doing different things. And the minimum then was a 2.6. So about half of the people said, I have no idea what you're talking about when you ask me this question. And about half of the class said, yeah, I probably could do some of it. So that's where we're starting off the semester. So there were only 45 people whose data I looked at, but that was the range, 1.4 to 2. And it turns out that when most of you, I just want you to know this is the reason that I'm spending the time talking to you about this in class, is because a lot of you, when I asked what are your fears about this class, you said either I've learned it before or I've tried this before and I'm not good at it. So I just wanted to let you know, if you were in the camp of the, I don't want to learn about that because I'm not good at it, or I've had it before and I had no idea what was going on, that when people said, for most of you, you said, I do not understand the task, when people rated themselves as 3s for any of the topics, it was sort of questions that had to do with probably vocabulary that we haven't defined yet. So epigenetics. Diagram how epigenetic modifications to molecules controls gene expression. We're going to talk about that in class, but we haven't talked about what epigenetics is. So I expect people to say, I don't know what you're talking about at this point in class. So if you were scoring yourselves 3s, that was usually why. And that's fine, because that's what we're going to talk about in class. We're going to go through it all. We've got plenty of time to do it. Everybody starts off on the same level, and I'm going to try to help build everybody up as much as I can with your all collective help. A couple other minor points. One thing that I just really wanted to point out, for those of you that have been watching these recorded class meetings, which I have been posting now, there are 2 of them on YouTube. So you can watch them before we get started. Oh, dear goodness. Anything at all? Wow. Is there a sound? Sometimes I'll think to turn the volume down there for you. Sorry for deafening you. The one thing I wanted you to know is if you do ever want to go back and look at any of this material again, you want to watch the whole 50 minutes, I'm sure. You want to relive the whole class because it's an outstanding experience. If you don't want to do that, for every one of these videos, after I go upload them, down here in the comments, I add a table of contents. So if you remember what you want to go back and review, but you don't want to watch the whole 50 minutes, go to YouTube and click on the, oh yeah, I wanted to remember about the analysis of HTLV nucleotide composition. Right, then it jumps you right there. Please don't watch the whole video if you don't need to. Make use of that table of contents if you're going to go review some of these videos. Okay. Now, on to business. I realized today that I do a horrible job of teaching this because I never start with what is a chromosome. I just assume everybody knows what a chromosome is, and many of you probably do. And so one of the traps about teaching any sort of chemistry, biology, science is I want to make sure that you don't get bogged down in vocabulary if I don't carefully define it. So does anybody have just a quick definition of what is a chromosome? Yeah.
It is a string of DNA that's been condensed into a form that is more easily transportable.
Okay, so I will agree with the DNA. Sometimes it's condensed, sometimes not. Is there anything else other than DNA that is part of a chromosome? Remember, DNA is A's, T's, G's, and C's, adenines, thymines, guanines, cytosines. There are proteins. So sometimes the DNA is condensed, sometimes not, but it is packaged by proteins. So a chromosome is both nucleic acid nucleotides, and it's also proteins. When you think of a chromosome, if you think of a chromosome, if you picture it in your mind, what do you picture?
Instructions?
So mental image is, so that's what DNA does. It provides instructions for making us.
What makes us?
What?
What makes us?
It's what makes us. But if you think about what a chromosome looks like, a ladder. Okay, so a double helix kind of looks like a ladder. And then if you look at a chromosome as in the chromosome, a lot of people think about an X-shaped thing. That's the type of, when we look at chromosomes under microscopes, that's often what we see. So this thing, and we're not going to spend much more time on this, this thing that is the X-shaped thing is a double helix, just zoomed way out. Right? Each of these arms is basically one double helix. So there are two double helices here next to each other, and that's what makes that X-shaped thing. So it's all nucleic acid, the blue stuff, and protein that's associated with the DNA, the double helix, that makes what we see under the microscope as a chromosome. How big are chromosomes? How do we normally measure DNA content? So base pair. So how many base pairs long? And remember, a base pair is one of these things. So A, T, G, C, and so forth. So how long is a human chromosome, for example? 10 base pairs, 100 base pairs, 1,000 base pairs, 10,000, 100,000.
Aren't they like 10,000?
They're like millions, tens of millions of base pairs long. Millions of nucleotides or millions of base pairs. So when we're talking about now humans, different organisms have chromosomes of different lengths. And some organisms have chromosomes that aren't even linear. What's an example of an organism that doesn't have a linear chromosome?
Bacteria.
Okay, so what shape? They're triangular? Yeah. So bacteria then have circular chromosomes. And we talked last time about a type of organism that doesn't have a double-stranded chromosome. It's got a linear single-stranded chromosome, some viruses. So chromosomes, double-stranded DNA, nucleic acids, plus proteins to help package them. And they're variable size. So when I talk about a chromosome, that's what I'm envisioning in my mind. Millions of nucleotides, millions of base pairs, often shaped like X's, although we're going to see today how that's sometimes not the case. Okay, so if you go into Google Classroom at this point, you should see that there's been a question posted. There's one comment so far, but I'd like you please to open Google Classroom, and again, our fabulous discovery guide is in the class today to help me with any technical issues. So if you go to Google Classroom, either the app or on the website, you'll see this question: what is the complementary sequence to this? And I'm just asking each one of you, please, to put in, as a comment, your answer. It'll take a couple minutes to do this. So while you're doing that, those of you that have finished, is this the type of question that has one right answer or multiple possible right answers?
One.
So anybody else?
I would say multiple.
So we're about to find out. So far, so good. So how would there be multiple? There's only one complement to this sequence. So how could there be multiple right answers? I tend to agree that this is a yes or no, right or wrong is factual question. We're building a portion of a chromosome right now.
So if mRNA was decoding it, then instead of T's, you would get U's?
So if you were base pairing this with RNAs, then you'd have U's instead of T's. That's an interesting point. True, absolutely. I'm just waiting to see, and it looks like, I'm going to show you, let's see. Everybody has written exactly the same thing, which is fantastic. I've only got one little quibble, and this is why I was doing this, because I wanted to start giving you feedback on, this is one of the lowest point questions I would ask on an exam, which hopefully you would appreciate because you've probably done this in many classes already. A lot of the answers I'm seeing are 3', and so everybody's remembering to label the polarity. That's one of the things I was looking for. I wanted to make sure everybody was going to label the polarity, not just write down a string of letters. That's good. So a lot of people are starting with 3' and then base pairing, T, A, so forth, down to 5'. That is the correct answer. So everybody, for the most part, it looks like, has done that. What's the one thing that could be better than this? It's a fine point, but it adheres to something that biologists and geneticists agree is a convention for writing DNA sequences. It's a simple problem, simple change. Yeah, flip this, because we usually write DNA 5' to 3'. Now, I understand why you do this, and the reason that I want to tell you this now, this is a fine point. In fact, it's so fine a point that I don't actually care. So if you did this on an exam, you'd get full points. I just wanted to make sure that everybody remembered that the orientation that we normally write DNA is 5' to 3'. This actually then turns out to be, for me, not a question that has one right answer. I'm flexible in the grading of this type of question because if you show me you're understanding what you're doing, even if you don't follow the convention. Okay? I'm flexible, in other words. As long as you show me that you're understanding and comprehending the material, then the grading is easy. Okay? So thanks for doing that. Any questions about this? Just to review. Making a quick point. All right. So we've got 3 tasks for today, all about the structure of chromosomes, how chromosomes look, and maybe eventually answer the question, why do chromosomes have stripes? So that's where we're heading both today and tomorrow, whatever we don't finish today. So one of the tasks was labeling centromeres and telomeres. What's a centromere? How do you know where a centromere is? Take multiple hands. Centromere is?
The place where the 2 strands touch.
The places where the 2 double helices touch, I'll clarify, but yeah. So there, if it's an X-shaped thing, it's the place where the 2 chromosomes, the sister chromatids, appear to touch or cross. Okay? Easy, maybe. Except, do all chromosomes have centromeres? What does the centromere do? We haven't taken cell biology yet necessarily, but there's something that is important about the centromere. Yes.
Bless you. When the cell is going from one cell to 2 cells, what are they called? The centromeres are attached and then pulled apart from that point, leading to having 2 identical sides of it instead of accidentally taking a bit that's not supposed to.
Exactly. So in short, the centromere is where microtubules grab the chromosomes and pull them into the 2 daughter cells at cell division. So every chromosome has to have a centromere. Because if you don't have a centromere, then you don't segregate properly. The chromosomes don't go correctly into both of the daughter cells. And that winds up doing things like something we'll talk about later in class, aneuploidy, meaning cells that don't have the correct number of chromosomes. What's a common human disease that comes out of aneuploidy? Wrong number of chromosomes. Down syndrome is probably the most famous. Got 1 too few chromosomes. Instead of 46, you've got 45, and then you get Down syndrome. Right, so centromeres are important. Every chromosome has them, has 1 or 2. So what about this little guy down here, or gal? Is there a centromere there?
Yes.
Where's the centromere?
Really close to the top.
Yeah, it's at the top. And if you watch the videos, you know that there are metacentric, submetacentric, acrocentric, and telocentric chromosomes, and this is just an example of some of each. We have metacentrics, where the centromere looks like it's about in the middle, from top to bottom. Submetacentrics, it's off to one side, sort of acrocentrics, it's near the end, and then the telocentrics, where this telomere looks like it's at the very tip of the chromosome. So how do you go from metaphase chromosome spread, which is you take a nucleus from an organism, you drop it on a microscope slide, it pops, and the chromosomes just lay there on the microscope slide. So that's how geneticists start looking at chromosomes. How do you get from there to, that's a metaphase spread, to a karyogram? That's this bit. What did a scientist do? What do you think? It's easy. What do you think? Well, this is actually light microscopy. It doesn't even require X-rays. You just look at them under a light microscope, a compound microscope. But literally, when I take a picture, this is not my picture, by the way. This is of a really crazy mammal called a muntjac. They have the smallest number of chromosomes of any mammal, which is why it's easy to look at pictures of them. It doesn't look too complicated. Somebody actually had that on a microscope slide and took a picture of it. What did they do? It's the answer that's so obvious, to get a picture that they published in a manuscript that looks like that. That's the karyogram. Sir?
Don't they just organize it by the size?
Literally. You'd cut this apart. It used to be actually you'd take scissors and print out the print, and you'd take scissors and cut out the different chromosomes, spin them around, and orient them so that you do, depending on the organism, largest to smallest, and then sometimes organize them by type of chromosome. Metaphases, largest to smallest, submetaphases, largest to smallest, and so forth. So what's an ideogram? That's karyos. This is a metaphase spread. This is when you organize them in some way visually, that makes a karyogram. And then the ideogram. Cartoon representations of all the chromosomes. So how many different chromosomes or objects should we see here? This is from a human. So humans have 23 different chromosomes, numbered 1 through 22, and then we've got the X or Y, X and Y. That's the 23rd type of chromosome. So that is what scientists, geneticists call N, the number of different chromosomes. Chromosome 1 is different from chromosome 2, is different from chromosome 3. So if this is human, I'm not a human clinical cytogeneticist, but since they number them from largest to smallest, I'm going to guess that that's chromosome 1, and that's the second copy of chromosome 1. And this is what cytogeneticists do, is then they say, well, maybe chromosome 2 is that submetacentric, sort of second largest chromosome. So we're diploid. We have 2 copies of each chromosome. Where did they come from? Mom and dad. So at fertilization, dad contributes 1 copy of every chromosome, 1 through 23. And mom contributes 1 through 23 in her egg. They come together, they form a single cell embryo that's got 2 copies of every chromosome. That's the 2n equals 46. So if you look in any cell, you should see that number, 46 objects. There are 2 copies of chromosome 1, there are 2 copies of chromosome 2, 2 copies of 3, 2 copies of everything through chromosome 22, and then there's either an X and an X or an X and a Y, depending on whether or not you're female or male. So is there a karyogram up here right now? What's a karyogram?
When they're organized.
When they're organized. This is just a metaphase chromosome spread. This is what the nucleus looked like when it popped on a microscope slide and all the chromosomes fell onto the slide and laid out like this. By the way, this is tough. Maybe especially if you're into genetic counseling or something, you might do this someday. It's actually hard to get all the chromosomes spread out so they're not all overlapping each other. So it's really hard to actually get a picture like this in the first place. And yeah, so to make a karyogram, then you'd cut these all out and rearrange them and make the nice image of all of them lined up. So the karyotype, then, is this 2n equals 46. It's the short sort of numerical description of the chromosomes you find in a cell. So here's a karyogram of a human male. It's got an X and a Y, differently sized, this last pair down here. So I know that's a male. But these chromosomes don't look like the chromosomes we were just looking at. What's different about this slide? The individual things, the 2 copies of chromosome 1, the 2 copies of chromosome 2, the 2 copies of chromosome 3, they don't look like X's this time. They just look like a single line. So we're going to talk about the difference between the two later. I just wanted to show you both versions because sometimes people get confused about looking at this where they don't look like X's and the previous slide where they do look like X's. But the take-home message is every time you see a single distinct object, be it just a line or an X-shaped thing, that's a metaphase chromosome. They can have different shapes, bottom line. We'll talk more about that in detail as the class proceeds. So these are 2 different things. This is the karyogram, the karyotype. Sorry, karyogram, unordered, metaphase spread, karyogram, and the karyotype is 2n equals 46. There's only one other thing to add in the case of humans to a formal karyogram is that you say what the sex chromosome constitution is, XY in this case. And the only reason you know that, and the only way you can tell, is the 2 X chromosomes, if you're a female. Not me, I'm just, anybody else? I'm XY. As far as I know, I've never actually been karyotyped, so I'm assuming I'm male, but remains to be seen. If you have 2 X chromosomes, they're 2 X chromosomes. They look the same. So if this was a female, the 2 large chromosomes would be identical in size, shape, and the other thing we haven't talked about yet, these aren't just shapes, they also have some variation. So what causes this variation in the shading of the chromosomes? It's distribution of something. It's not distribution of proteins. It's something else that we'll probably get to either later this class or tomorrow. But these bands, if you just look at a chromosome, I'll get to you in just a second. If you just look at a chromosome under a microscope, it doesn't look like that. It doesn't have variation in shading. It just looks like DNA. So there's something that scientists do to elicit, to bring out these patterns. They add specific chemicals that react differently to different nucleotides. And that's how this shading pattern is revealed, essentially. So this is not this shading, the bands, the dark bands, the light bands, the gray, the white, that's not a characteristic of a chromosome in itself. That's something that chemists do after they get a microscope slide with chromosomes on it. And the only reason, well, why do we care about chromosome banding? What makes it useful? If you wanted to know which of these chromosomes is chromosome 8, how would you know? Well, you could go 1, 2, 3, 4, 5, 6, 7, 8. That's the 8th chromosome. But if you were going to try to tell somebody else, this is how you find the 8th chromosome, chromosome 8 in humans, how would you describe the chromosome to them? Well, you look at a nucleus and you find the, well, shoot. You look at all the, well, in the back, pardon? So you could actually sequence all the chromosomes. That's a lot of work and a lot of expense, though.
After you treat the chromosomes with that, what makes it banded, it's more easy to tell which ones match because they have the same density of that DNA in the same areas,
Okay.
have the same genes on them.
Okay, so we've got 2 different pieces of information, in other words, that we can use to sort chromosomes or identify chromosomes. One is their size, their length relative to each other. The other one is each of these banding patterns is a fingerprint of a chromosome, essentially. Each chromosome will have a different banding pattern. So if you get the banding patterns, you can see that those 2 chromosomes have equivalent banding patterns. They look the same. They're not just the same length, because those are about as long as those ones. So you have the length, but also to really clarify which chromosomes are which, we can stain the chromosomes and look at banding patterns. And that's the only way for most organisms you can actually tell which 2 things over here, not banded, which pairs, if I asked you to pair this up on an exam, well, that one kind of looks like that one, but maybe it's that one instead. So you have to have banding. It's not a property of the chromosomes, it's something that we have to do to treat the chromosomes to see the banding. getting back to the difference in shape between the linear chromosomes, the ones that look like sticks, and the ones that look like X's, that is entirely to do with which stage of the cell cycle the cell was in when you broke it open and looked at its chromosomes. It's the only difference. So if you look early in mitosis or meiosis, which we'll spend more time in class on later, it's going to look like the two double helices are stuck together all the way down their lengths. And that's done using a type of protein called a cohesin, these little red dots, physically glue the two double helices to each other. So it doesn't look like an X, it looks like two pieces of double helix that are stuck all the way along their lengths. But then as cell cycle proceeds, most of those cohesins break, and then the two arms can spread apart, and then they look like X-shaped things. So they're all chromosomes. But the only stage, the critical thing to take away from this, is the only stage of the cell cycle when we can actually see chromosomes, please remember this, is metaphase. When, as was suggested earlier, the chromosomes are condensed. DNA is usually really like, it's like spaghetti. It's just all over the place in the nucleus. It's not particularly organized. That's kind of a lie, but not really. It's diffuse. It's like cotton candy in the nucleus. It's fluffy and spread out. And at metaphase, then all the chromosomes condense. And that's the only time we can use microscopy to look at chromosomes. So if you ever see a microscope slide that's got chromosomes on it, it's a metaphase chromosome spread. I'll let you do this on your own if you want to, not now. If you want to get the answers to this, I'll have the answers available. Just another practice. This is one from the manual, if I remember correctly. So you can look up the definitions of those, and if you want more help with that, come see me. You don't need to know this for exams. I just wanted to show you, a lot of students sometimes ask, well, how do I know if it's metaphase or submetaphase? Because, if the centromeres kind of off, it's definitely not metacentric because the centromere is not in the middle of the chromosomes, so if I ask you to classify chromosomes on an exam, what am I to do? Is that submetacentric? Is it acrocentric? So there is actually, published in 1964, this really fun paper that numerically describes how you classify chromosomes into submetacentric, acrocentric, telocentric. You don't need to know that. I just wanted you to know that if you really are that objective about this, you could do this. Basically what it means is, if the chromosome was shaped like an X, where that centromere is, if it's in, then you divide the half of the chromosome in quarters, physically. And so which of those intervals the centromere is in then defines whether it's metacentric, submetacentric, acrocentric, or telocentric. So if it's anywhere in the first quartile of the center, that would be metacentric, then submetacentric. If it was up here, it was acrocentric, and if the centromere is way at the end, then it's telocentric. So if you really want to know the details, those are the details. Your turn. Feel free to talk to a neighbor if you want, or don't, but I'm happy to let you have a second to do this. What's the discrepancy? There's something wrong here. There's a karyogram and a karyotype. So in 2 minutes, I'd like you to be able to tell me what the problem is. Go ahead. Questions? Okay, what's the issue, if there is one?
Is that a monosome?
Pardon?
Is that the right term, monosome, instead of trisomy?
So it does look monosomic. Good use of terminology we haven't gotten to yet, but yeah, monosomy means it's got, what does that mean? What's wrong with the karyogram and the karyotype?
2n equals 6, there should be 6, there's only 5.
Okay, so what I heard was, this says that this is an organism that's 2n equals 6. So how many chromosome-shaped things should there be in a metaphase chromosome spread? There should be 6. There are 3 different types of chromosomes, you're right. So if we labeled these, if this was a human and we were going to number the chromosomes, which chromosome number would something that size be, probably? Is that the biggest chromosome? Is it the smallest chromosome? Okay, so we're going to call that chromosome 2. Does it have a partner? Is there another chromosome 2 up there? Something that's the same size, same shape. I would say maybe that one. Okay. Is there what's chromosome number 1? Where are they located up here? The bottom and top right, maybe, about the same size. Looks like the centromere is somewhere about in the middle of the chromosome. Okay. So we'll call this chromosome 3. So monosomy. This cell only has one version of chromosome 3 in it. There should be something else here that has the same shape and approximate size. That would be 2n equals 6. What we have right now is 2n equals 6 minus the one that's missing. 2n equals 5. So that's a karyotype for not for monosomy. Well, it's monosomy for chromosome 3. There's one copy, monosomy, one body. Good. Skip over a few of these. Actually, no, go ahead. We got time. So if this was a metaphase chromosome spread, it's ideograms. They're cartoons, not real chromosomes. Think about it for half a minute. Karyotype. How's the karyotype going to start? Okay, who says 2n? Let's do show of hands. The only thing we've seen so far in class is 2n. And that's just today. We haven't gone very far. Do you know what, I've already said this term once, we haven't talked about it in detail. Do you know what the other word for saying something is 2n, 2 copies of every chromosome? Diploid. Fantastic. Are all organisms diploid?
No.
Not every species on the planet has 2 copies of every chromosome. What's a big category of organisms that sometimes has a wide variety of the numbers of chromosomes they have?
Salmon?
Pardon?
Salmon?
Salmon and plants. That's impressive. Not most people know that salmon sometimes have 8 or 12 copies of every chromosome. Plants are notorious for having multiple copies of chromosomes. Some strawberries are dodecaploid. They have 12 copies of every chromosome. So if they had 3 chromosomes, they'd have 36 double helices in their cells. I don't know why plants need so many copies of every chromosome. I mean, 2 does pretty well for us, I think, but they're strawberries, so who knows? Yeah, too bad, plants can't talk to us. So 2n equals what in this drawing? How many, so there are 5 different types of chromosomes. So n equals 5. If you look at the ideograms, you'll see that there are 5 different banding patterns. 5 chromosomes have 5 different places where their centromeres are. So that means that 2n equals 10. There are 10 different objects. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Wow, yellow shows up so well. Sorry for that. And if you don't believe me, we're not going through this, but if you don't believe me, this is the comparison. So that's the banding pattern of each of the 5 chromosomes that this organism has. And twice that number at metaphase is 10. Okay. Because I'm going to ask you to be looking up primary literature, manuscripts on your own, it's worth taking a little bit more time to see if there's any questions you have about 2 different things. One of which is looking up references using PubMed or whatever your tool of choice is, Google Scholar Search. The other thing is citations. Does anybody have any questions about what content is in a citation? Some of the things we're going to do in class is going to require you to understand what exactly all of this 2 lines of text means. If you're going to be scientists and you're going to look up literature and read it, question?
The very last part after the publication.
So this bit.
Yes.
Yeah, exactly. Thank you. I'm glad you were willing to suggest that. That was exactly where I was when I was in your seats. It's like, I understand, so first part, the authors come first. So it's somebody's last name. Et al. Is Latin for and others. So it just means there were more authors, but I didn't want to write them all down. Lazy. The year it was published, the title of the paper. Okay, straightforward. In italics, does anybody know what this is? This is the name of the journal. Like magazine. Like Better Homes and Gardens, or Discover, or Scientific American, or Fit and Healthy, I don't know. So that's the physical thing that you can pick up in the library or go to a website for to look for this manuscript. Bold number, volume. That is like some magazines, every year they have a different volume. So in 2019 might be volume 36. The next year they say all of the papers, all the physical magazines we publish are going to be volume 37. So the rest of this, the numbers are to more finely direct you from what publication, what journal, what thing do I look in to which version of that journal, the volume. Sometimes there will be an issue number, like month maybe. If it's a monthly journal, then there will be journal issue 1 through issue 12, and then the page numbers. Or, in the case of electronic formats, a single number that's a unique reference for a specific paper, like E254. So what are you going to do when you need to look this paper up? Because this is actually, the diploid genome sequence of an individual human is a paper that we're going to look at in class. And I'm not going to put it on Canvas. So what are you going to do to find it? What's your best bet? Okay, use PubMed. What's even easier than that?
Put that directly into Google?
Damn right. Just open Google and Google the diploid genome sequence of an individual human. Don't bother with the authors. Don't bother with the mundane details about where exactly this paper is published. More often than not, PubMed will probably come up as one of the top hits, as well as other places. The only reason that I stress PubMed is that, as you saw in the video, if you watched it, one of the shorter ones for today, that PubMed will tell you if it's freely available, and we'll give you the link to the freely available version, so you don't have to log into the Henry Madden Library website and go from there to try to find the journal, find the issue, find the page range that contains this journal. So everything that I'm asking you to access this semester is able to be downloaded as a PDF for free. So you shouldn't, if you've gone somewhere where it's asking you to pay $300 for a 24-hour access, there's a free way to get all of these papers. Don't pay any money. You've already paid enough. Okay, at this point, I'm going to skip to, so we'll cover the rest of all of these slides next time, except I just want to end with a few points. So first thing is there's going to be a Socrative in just a minute. Okay. So go for that. And then once most of you or all of you are in, then I'm going to tell you what we're up to next class, which is finishing up what was in the PowerPoint for today. I will upload a new set of PowerPoint slides or PDF of them for next class, but they'll be mostly the same as what you haven't seen yet today. But we will finish this by the end of next class, so there is the next chapter, genomes, to look at for the class after next. Not next class, not Wednesday, but for Friday we'll move on to chapter 1.3, the genomes chapter. So I should say, for Friday.